Alexandra Harryman, who was a Meyerhoff student, cohort M6, while at MA, my eyes are failing me, MA, while at UMBC as an undergraduate, is now back with us as a graduate student and is supported by Meyerhoff Foundation as a PhD student. And Alex's talk is entitled Investigating the Roles of Reg A and Other Varl Genes in Cellular Differentiation in Volvox Carteri.
All right, so good morning. As Steve mentioned, I was an M8, and I'm currently a fourth-year student in his lab at UMBC. And the title of my talk is Investigating the Roles of Reg A and Other Varl Genes in Cellular Differentiation in Volvox Carteri. So one of the main questions that our lab seeks to answer is, how did multicellularity arise? In other words, how does one cell type give rise to two different cell types? And as you can see, multicellularity has arisen in several lineages. Most notably, the eukaryotes in plants and animals, this transition occurred between 450 and 700 million years ago. However, we use an organism with a much simpler developmental system in order to study the molecular framework that led to this transition, that allowed for this transition. And that organism is the green alga Volvox carteri. And Volvox carteri lends itself as a good model organism for several reasons. First of all, it contains only two cell types. So what you see here are the larger cells, which we call ganidia, and they're responsible for reproduction. And Volvox also contains approximately 2,000 smaller cells that are responsible for motility, and they're terminally differentiated, and we call them somatic cells. Volvox also reproduces rapidly. Its asexual life cycle takes about 2 days. So to give you an overview of this life cycle, as will be pertinent to the research I'm presenting, you begin with the parental spheroid, which contains approximately 16 of these ganidia reproductive cells. And these cells go through a series of symmetric and asymmetric divisions to produce what we call juvenile spheroids. And then these juveniles undergo an inversion process and a cytodifferentiation process. And it's this stage that our lab is most interested in, because it's at this time point that presumptive somatic cells will develop eye spots and flagella and adopt a somatic role, and the presumptive ganidia cells will become reproductive. So these juveniles continue to grow and expand until they're released from the parental spheroid to begin this cycle all over again. So another of the reasons that Volvox is a good model organism for our studies is because it belongs to a lineage that contains members that display a range of developmental programs. So I need to say that this schematic here in no way implies a direct evolutionary relationship. It's simply meant to show this range of developmental phenotypes that you see among the members of the Volvocene lineage. So on your left here is Chlamydomonas, and its unicell first serves in motility, and then it differentiates into a reproductive cell. And at the other end of the spectrum, you see the multicellular Volvox, which sets apart the two cell types through a series of asymmetric divisions, and then segregates these two functions, reproduction and motility, into two cell types. So Chlamydomonas and Volvox diverged from the unicellular ancestor approximately 50 million years ago, which is much more recent than the transition to multicellularity that we see in plants and animals. So this allows us to better tease apart the molecular framework that led to this transition. In addition, we have a variety of molecular tools available that we use at our disposal to study Volvox. And one of these tools is for genetics, in which we can identify, which has allowed us to identify key genes in Volvox development. So what you see here is one of the phenotypic or developmental aberrations that we see, and it's called the Reg phenotype. And these Reg mutants will grow and differentiate normally during the first day of development, but then during the second day, the somatic cells will undergo a dedifferentiation process where they lose their eye spots and flagella, and then they enlarge and assume a reproductive function. So this is sort of reminiscent of what we consider to be an ancestral developmental program in which cells are first biflagellate and then reproductive. So since we have identified the gene that's responsible for this phenotype, and that's Reg A for somatic regenerator. And we believe that Reg A has evolved in Volvox, and what it's allowed it to do is take a certain cell type and set apart that cell type for a particular function, in this case, the somatic function. Okay, and I need to say, sorry. Since then, molecular analysis has also allowed us to determine that the Reg A protein is expressed exclusively in somatic cells, It's nuclear localized, and it contains a number of transcription a number of motifs that are common to transcription factors. And the one that we're most interested in is what we call the Varl domain for Volvoxine alpha Rega. And the Varl domain is of interest to us because its secondary structure bears resemblance to the secondary structure of the SAN DNA binding domain that's found in a number of plant and animal transcription factors. Furthermore, there are 12 other genes in addition to Rega and Volvox, and 12 genes in Chlamydomonas that contain this Varl domain, and we call these genes RLS genes for Rega-like sequence. And the ones that my research is interested in is RLS-D, because the Varl domain of RLS-D is most similar to that of Rega, and RLS-D also contains an ortholog in Chlamydomonas, RLS-1. Secondly, RLS A through C belong to what we call a Reg A gene cluster. So RLS A through C are paralogs of Reg A, and this expansion of these Varl genes that you see here is unique to Volvox Carteri. So we've developed some hypotheses as to the function of these Varl genes based on this, and we believe that because RLS A through C are paralogs of Reg A, that they serve a similar function in specifying and maintaining the somatic cell fate in Volvox Carteri. So one of the main goals of my research is to perform structural and functional analyses of these RLS genes, and RT-PCR has told us that these genes are expressed in Volvox. And last summer I had an opportunity to work with one of our collaborating labs in Japan to begin expression analysis of these genes. So what I did was I performed real-time PCR. Real-time PCR is a method that allows you to determine the expression level of any given gene or at any time point during development of an organism. So to orient you to this graph, you're seeing here real-time or relative expression levels of Reg A, RLS-B, and RLS-C. And the boxes here sort of represent the cells that were isolated and assayed to get this data. So the data here is representative of the expression of these genes in only Ganidia, or the reproductive cells. Here it's representative of expression in presumptive, somatic, and Ganidia, and the latter part of the graph shows the expression in juvenile, somatic, and Ganidia. At these time points here, it's very difficult to separate these two cell types at these stages during development. So what you see is that Reg A is not expressed in reproductive cells. Its expression is exclusive to the somatic cells, and the highest point of its expression occurs at the time that cytodifferentiation begins. The same can be said for RLS-B and RLS-C, which also show a similar pattern of expression, and also are not expressed in the reproductive cells, we believe. Furthermore, RLS-B and RLS-C are expressed during a slightly narrower window during development. However, if we add RLS-D to this picture, the story becomes a little bit different. So RLS-D, like RLS-B and RLS-C, is most highly expressed when cytodifferentiation begins. However, its expression is much more variable throughout the entire life cycle of Volvox. In addition, and probably one of the major differences, is also that RLS-D seems to be expressed in the reproductive cells of Volvox Carteri. So we focused our attention on RLS-D and made some hypotheses as to its function. And so there's a previous research that has shown that RLS-1 expression is upregulated in Chlamydomonas when it's grown in unfavorable growth conditions. So because RLS-D is an ortholog of RLS-1, we believe that it serves a similar function in Volvox, and its expression also may be upregulated when Volvox is grown under stress conditions. So the next series of experiments that I performed were to test this hypothesis. So what I did was I grew Volvox either in the absence of light or in the absence of sulfate, and I looked at the relative expression levels of these Varl genes again. And I need to say here that this data set is not, I won't say incomplete, but I am working on reproducing this data, and this time including proper housekeeping controls. But what is evident here is that you see that the expression levels of Reg A, RLS-B, and RLS-C are almost completely diminished when Volvox is grown under these stress conditions. But again, if we add RLS-D, we see that this gene is not downregulated nearly as much as these other Varl genes. So this suggests to us that RLS-D has an important function during this time in Volvox, or during Volvox development when it's grown in unfavorable conditions. So to revisit, I guess, our working hypothesis, so we believe that the progenitor of RLS-D and RLS-1 in the unicellular ancestor of Volvox and Chlammy was responsible for suppressing reproduction conditionally when growth conditions were unfavorable. So Reg A and RLS genes co-opt to this function. However, these genes are constitutively expressed, and they're expressed exclusively in one cell type to establish and maintain somatic cell fate. This also sort of leads to a hypothesis that these genes the molecular framework of these genes were always present in if not the entire Volvox lineage, at least Volvox and perhaps Chlamydomonas. And it was just an adjustment of the temporal and spatial timing which allowed for this multicellularity to evolve. So the future directions of my research, as I said, I'd like to complete the real-time PCR analysis of RLS gene expression. I need to include data from the parental somatic cells. I'd like to finish my structural analysis of these genes and use northern blots to determine the transcript size. And finally, I'd like to knock down RLSD and RLSB and then look at, I guess, the effect on the development of Volvox to start making some guesses as to their specific functions in Volvox development. So finally, there's a lot of people I need to thank. First of all, I'd like to thank Meyerhoff for allowing me to speak today. I'd like to thank our lab, Dr. Miller, our moderator. The most recent, I guess, well, she's not a graduate, but Dr. Valeria Pappas, who just recently defended her thesis. And the other two members of what we call the A team in our lab, Alicia and Akelia. I'd also like to thank the Nishi lab and the wonderful group of postdocs that I worked with last summer that were instrumental in helping me to get this data. And I'd like to acknowledge my funding sources, Meyerhoff, the U.S. Department of Education, and then finally National Science Foundation, National Institute of Health, and the Japanese Society for the Promotion of Science for funding my research last summer, which I presented to you. Thank you very much for your questions.
Okay, I think we also have time for one question. Yes.
Before you began your comments on the real-time PCR results, you referred to one gene or one gene group as being unique to Volvox cellular. I was just wondering exactly what that meant.
So what I was referring to was what we call the Reg A cluster, and so what you see is I guess a tandem array of genes, and it's RLSA, RLSB, and RLSC. And RLSA, B, and C, I guess we consider that as expansion genes because they're paralogs of Reg A that arose through duplication. But Volvox is the only, we don't see that in Chlamydomonas. So Chlamydomonas, we don't think that any of these genes have duplicated. So we believe.
But by using the term unique, you didn't mean that it couldn't be found in, for example, other species of Volvox, etc. Just in this comparison.
It's in, as of now, yes. We don't know if these genes have been expanded in other Volvocene and Volvocene.
Okay, thanks very much.